Hello students, this video is to help you understand how to do your assignment on GoFormative for the mutations. So I'm going to be doing an example with you that is essentially the same thing that you'll be doing in your own assignment. So you'll be given a sequence of DNA and then a sequence of mutated DNA. And the first thing the question asks you to do is to transcribe and translate the DNA. So when we ask for transcription, that basically means you need to change it into mRNA. So remember that C's always pair up with G's, A's always pair up with U's, T's and A's. So all of this is going to look the same, GUA, GUA. And then the second thing I need you to do is then look at your codon chart to translate it into its amino acids. So since remember that a codon is made up of 3 letters, so three bases, and since it's all GUA, 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 it'll be pretty easy. If you look it up, all of these are just going to code for valine. So val, 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 and val. Okay, so we've got 7 vowels there. And in your second sequence, so now that you have your mutated DNA, you're going to do the same thing. You need to transcribe it and translate it. Now be careful because you'll see that it is almost exactly the same as the original DNA, but you'll see that some of these sequences have a change in them. So now I ran into my first change here. Instead of CAT, I have CCT. So this is going to be GGA, and then it looks like I'm back to GUA, GUA. So this time when you go and translate it, okay, so my first one, I have val, 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 and then GGA. If you actually look that up on your codon chart, it translates into glycine, GLY. And then after here, everything is back to normal again. So we've got val and val. And then I think the next question it asks you to do is to circle where the DNA got mutated, where the DNA got changed from the original one. So if you look carefully, we have cat, 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 cat 7 times, and then in the second sequence we have cat, 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 and then it's C, CT instead of A. So there's our change right there. So the next thing you're asked to do is to figure out what type of mutation happened. Now you learned about this in the Edpuzzle video you watched, but really if you just think about what these words mean in regular English, it should make sense to you. So for the first type, a substitution or a point mutation. A substitution is when we just switch out or substitute one letter for another letter, just one single letter. In an insertion mutation, what's happening is like if it was cat, cat, and then we got like a thrown in there, and then it's cat, cat, and so on. Then we have an insertion. We got this extra letter added in right here. So it causes all of the rest of your sentence to get pushed down, and it messes up what the words are supposed to be. For a deletion mutation, it's just like how it sounds. Deletion is when we erase or delete a letter out. So let's say we are having, cat, cat, and then instead of cat again, we erase the C, and then it ends up being A, T, and then we're back to cat, 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 and whatnot. But since we erased a letter right here, Now the sentence doesn't make sense again. We have cat, cat, ATC, 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 so on and so on. So the insertion and deletion mutations are really bad because it causes all of your letters to move around. A substitution, when we just substitute or switch out one letter for the other, it might not cause a problem because it might just end up coding for the same amino acid anyway. So in our example here with the cat and CCT, here's where the change happened, and that looks like it was just a substitution. Instead of an A, we swapped it out for a C. So the answer here is A in this example. And then the last part that you guys are looking at is trying to figure out what effect does the mutation have on the protein that is made. So if we look at the protein that's being made in the fourth codon right here, instead of making val, we made gly. So that means we are making the wrong protein, which would be A, which is called missense. We made the incorrect amino acid, and we might be making a different protein altogether. We might make a bad one. In the examples that you're going to be doing, you might see missense. Nonsense is when we get an incorrect sequence causing shortening of the protein. So for example, let's pretend that instead of like translating into glycine, it translated into stop. And if it did that, then your protein would only be 3 amino acids long when it was supposed to be 7 amino acids long. So that means the protein is cut short, and so that is when we call it nonsense. And then you can also have the silent mutation where there's no effect. So basically, let's just pretend, you had all this, you had all your sequences going on, and the mutation, instead of coding for val, it just coded, it was a letter change, but it coded for val anyway. Well, then that would be a silent mutation. So this is the way you should be doing this assignment. So again, you're supposed to transcribe, translate, and then don't forget to circle where the error was or where the mutation was, and then answer the 2 questions afterwards. If you have any questions, then please email me, send me a remind, hit me up on the Zoom meeting. Hope you guys are doing well, and I'll talk to you later.